Hi, and welcome to this run-through for the 2018 biological process paper from the OCR A-Level Biology board. So you can find this paper at the link. I would recommend do the paper and then watch this video and mark it and see how you go. Or if you want, you can kind of do it with me. You can pause every once in a while, do your questions, and then see if you get it right. So without further ado, let's get going. Okay, question one. Which of the following statements, A to D, correctly explains a feature of an efficient gaseous exchange surface? So let's look at each one. The layers are thin for a short diffusion distance. Yeah, that's true. There is a good blood supply, so the system reaches equilibrium quickly. I'm going to think about that one for a second. I'm going to come back because there's only one which is correct, so it's probably this one. C, there is an increased surface area to reduce surface area to volume ratio. No, that's not correct. We want to increase the surface area to volume ratio. Ventilation takes place to reduce concentration gradient. No, we want to keep that high. I guess reaches equilibrium quickly. I guess we never really reach equilibrium in all gaseous exchange systems. We want to keep absorbing gases and getting rid of them. So I guess B is wrong. So A is definitely the best answer. So we go with A. All right, 2. The following is a series of organic molecules and the chemical processes that occur to convert them into different molecules. Which is correct? Okay, nucleic acid, like DNA or RNA, hydrolysis would take it to a nucleotide. Yes, that's true. That bit is true, sorry. Nucleotide to polynucleotide, no, that's polymerization. Alpha glucose condensation to amylopectin. Yes, that's true. And then hydrolysis would take it back to alpha glucose. Yes, that's true. So I think B is right, but let's just look at the other ones. Amino acids condensation would take to a dipeptide. Hydrolysis would not take it to a polypeptide. Beta glucose condensation to cellulose. No, this is not true there. So yeah, it is just B. That is fully correct. B. On 3. The following table describes the approximate percentage mass of different chemical elements in organic polymers. Ooh, which is correct? All right, well, let's look at the nitrogens here, okay? So, first of all, this is annoying because this is right. Nitrogen just is in nucleic acids and proteins. I think 30% is a bit high, I think. So I'm thinking it's A, but let's look at the rest of them. All right, well, let's look at the oxygens. Oxygen, 20%, 30, 10, 49. Okay, this is wrong. There's definitely not that much oxygen in lipid. It's more hydrogen. So hydrogen should be the highest for lipid, not carbohydrate, so I think that's wrong. Phosphorus. Okay, there isn't that much phosphorus in protein. 50, that's way wrong. So I think that's right. So yeah, I mean, you don't have to know these. I think I've got there by process of elimination. It's A, and that is correct. The main things there is that only nucleic acid and proteins have nitrogen, so actually all of this is correct. But then phosphorus is really just in, the sugar phosphate backbone of nucleic acid. It's not really present in protein. I mean, that is correct, but I think that's wrong. That is correct. What, could this be right? No, yeah, it's definitely A. And that's what the exam board says as well. So yeah, A. Moving on. 4. Which of the following statements, A to D, about DNA replication is correct? DNA replication. DNA into more DNA. RNA will bind to DNA through complementary base pairing. Now, this is talking about transcription. The distance between the strands in the double helix will always be the same. Is that really talking about DNA replication? I mean, that's true, but I don't know if that's talking about DNA replication. The proportion of adenine in a nucleic acid will always equal the proportion of guanine. No, that's not true. A binds to T, not to G. So A to T would be the same, not A to G. The formation of phosphodiester bonds will occur in the same direction on each strand during replication. That is true, because one strand is continuous and the other is not continuous. I don't think this is this. So I'm choosing between B and D, and I think B is not really about replication. It's just a fact. So I don't think that's really about replication. I think this is correct. This is about the process of replication. Yeah. Okay, and that is what the example says. Moving on, question 5. The following graph shows the rate of reaction of an enzyme in different substrate concentrations. Okay, A, B, C, D, all right. Okay, so we can see some are kind of flattening out there. And this one's flattening out at a lower level. Okay, which letter A to D shows the rate of reaction with a fixed quantity of competitive inhibitor? Okay, so with a competitive inhibitor, we can remove the effect of the inhibition if we increase substrate concentration. So we're looking for a line that approaches the maximum rate, but just more slowly than another line. So A is without anything, without any inhibitor, and B is with competitive inhibitor because it is slowed down, but if we increase the substrate, it will approach and eventually equal that maximum rate. It just takes a bit longer to get there. B. C and D would be with a non-competitive inhibitor, and D would be with this sort of more non-competitive inhibitor. And this A is sort of no inhibitor. Good question here. Got to learn these graphs, by the way. Should recognize those sort of straight away. Okay, moving on to 6. Which of the statements, A to D, applies to transpiration and evaporation? Okay. It occurs at a faster rate at higher humidity, transpiration, and evaporation. No. Higher humidity would slow it down, in fact. It occurs at a slower rate at greater wind speed. No. Greater wind speed would speed it up. It occurs at a slower rate at high temperature. No. Again. High temperature would speed it up. It occurs at the surface of leaves. I guess so. Yeah, it does. 6D. All right, question 7. Which of the statements A to D explains why diastole follows systole in the mammalian heart? So, what do these things mean? Diastole is the relaxation kind of phase, whereas systole is the contraction phase. So why does diastole follow systole? Cardiac muscle is myogenic. That's true, but it doesn't explain it. That is just saying that the heart can generate its own rhythm. B, cardiac muscle takes a short time to repolarize after being stimulated. That is true, and it also explains the statement. So B is the correct one, I think, but let's check the other two. C, the aorta is capable of maintaining the pressure generated by the left ventricle. That's true. It's a true statement, but it doesn't explain this. That's more to do with the aorta, being tough, lots of collagen fibers, slightly, sort of thick tunic of media, that sort of stuff. D, the SAN receives impulses from the AVN. It's actually the other way around, so that's false, and it also doesn't explain. So the answer is B. Okay, moving on. 8. Ooh, what's this? Looks complex. Okay, Banting and Best. Okay, these were the guys who did insulin, who sort of started to think about insulin for treatment of diabetes. Okay, it says, in one experiment, they removed the pancreas of dogs. Shortly afterwards, the dogs developed the symptoms of diabetes. Banting ground up the removed pancreas to produce an extract. He called the extract islatin. The islatin was then injected into dogs that had had their pancreas removed, and he tested the blood glucose concentration. Okay, we can see that after the injection of islatin, the blood glucose concentration goes down generally. Which of the following statements correctly explains these results? Islatin is made in the alpha cells of the islets of Langerhans. Well, that's false. It's actually the beta cells, and actually we wouldn't be able to tell that from this experiment. There's no knowledge about alpha and beta cells in this experiment. Islatin reduces blood glucose concentration. That appears to be proved roughly by this injection. Certainly, This one's a bit weird, actually. I'm a little bit wondering about this because it looks like the blood glucose concentration is already dropping prior to this injection, so I don't know why that is. It does appear the effects of isotin are short-lived because after the injection, it kind of takes the blood glucose concentration down just for a short period of time, and it comes back up again. So I would say 2 and 3, so therefore the answer is C. Yeah, and that is what the exam board says. Okay, moving on, 9. Ooh, another good graph to look at. Peak expiratory flow is a measure of the maximum rate at which a person can exhale. So that's like, sometimes if you've ever been to the doctor and they measure this, you have a thing, you blow in, and you just blow out as hard as you can. And this is called this peak flow rate thing. Okay, so this is the peak flow graph. Typical PEF values for men of different ages and heights. So let's just make sure we underline that. Men, different ages and heights. Which of the following is the percentage increase from the PEF of a 20-year-old man of 175 centimeters? Well, let's look at that one first, okay? And then let's take another colour for PEF of a 45-year-old man of 183. Okay, PEF, 20-year-old man, 175. Okay, so this 175 is the middle line. A 20-year-old man is here. So the middle line is there. Right there. And then, let's actually zoom in a bit. And then the other one is a 45-year-old man of 183. So 183 is the second to top line, and 45-year-old man is there. So we want the percentage increase from blue to red. So what are the numbers? This red line is 570. And this blue up here is 640. So what is the increase from there to there? So the change, I need my calculator. Can I do that? And I can do it in my head. So that's 30 plus 40. So the change is 70 plus 70. And remember, percentage change is the change divided by the starting value. So it's 70 divided by 570 times 100 to make it a percent. And that should be 70 divided by 570 equals times 100 equals 12.2%, 12.28. So that is C. 12.3 is the answer. And that's what the exam board says. Tricky graph reading there. Okay, zoom out again. Okay, question 10. One treatment for thyroid cancer is radioactive iodine. The radioactive isotope I-131 is used. The thyroid gland absorbs any iodine that enters the body, so the radioactive isotope kills the cancerous cells in the thyroid gland. The I-131 is then excreted from the body. Different body fluids excrete different proportions of I-131 as shown in the following graph. Okay, so most of it's coming out in the urine. Which of the following A to D correctly explains the different portions of I-131 in each body? I think A, soluble in water, would explain quite a lot of it. I-131 is able to cross capillary walls. Maybe. The kidneys are more efficient excreting I-131 than the lungs. Yeah, I think maybe more so because kidneys produce urine. The thyroid gland is well supplied with blood. I don't think that's it. I think C is the most correct there. Yeah, the exam board says correct, yeah. Because the kidneys produce urine. So C is right. Moving on. 11 and 12. Just moving a teeny bit more. Okay, 11. Which of the following A to D is a similarity in the way ATP is made in respiration and photosynthesis? Both involve NAD. No, that's not true. One uses NAD, one uses NADP. Both involve substrate level phosphorylation. No, that's not true. Both involve photons. Ooh, this is trying to trick you. No. Both involve proton gradients. Yes, that's true. So the answer is D. Photon is a particle of light, so that's just photosynthesis. Don't confuse photons with protons. 12. Diagram of the human brain. Which of the labelled regions would be directly involved in learning to play a musical instrument? Well, definitely Z, because that is the cerebral cortex. And the other answer would be W, because that is the cerebellum. And the cerebellum is all about control of fine sort of motor movements. So Z and W, and the answer therefore is C. Okay, 13. Which of the following diagrams, A to D, shows a sensory neuron? Okay, the answer is D. Now, how do I know that? The sensory neuron is the only one which has the cell body kind of outside of the main pathway of the impulse. So let's imagine the impulse is moving this way. Could be going the other way, we don't really know, but this is the sort of dendrons, and this would be this is the axon and the branching axon over there, and the cell body is to the side. This one, just so you know, this is a motor neuron, and the impulse is traveling this way. This one is a relay neuron found in the CNS. I'm not sure if that one really exists, to be honest. I think that might be something designed to confuse you. So 13, the answer is D. Okay, on to the last two multiple choice. 14. The table below shows the membrane potential of different neurons at a cholinergic synapse. Okay, so acetylcholine synapse. The data were recorded on 5 separate occasions, as shown in 5 rows. Which of the following, A to D, explains the data? 5 separate occasions. Presynaptic neuron. This is the postsynaptic neuron. Okay, I got you. So there's 3 different neurons, A, B, and C, all converging on one postsynaptic neuron. So this is really about summation. But what type of summation? Is it spatial or is it temporal? I think it's going to be spatial, but I'm just going to have a look at the data here. Here we've got, this one has basically fired. This is an action potential. This is not fired. This is not fired. And this is not fired. This is not fired, fired, not fired, not fired, or resting. Okay, so not fired, not fired, fired, not fired. This one is fired, and not fired, and still not fired. It's only when the 3, 1, 2, 3 are all firing at the same time, the postsynaptic one also depolarizes. Okay, so this is therefore spatial summation, because it's all happening at the same time. You know, this is 5 separate occasions, so therefore spatial summation is the answer. That's a tricky one. I haven't seen a question like that before. I like it. 15. The drug metoprolol prevents stimulation of postsynaptic receptors in the sympathetic nervous system. Which of the following conditions could this drug be used to treat? In the sympathetic nervous system. Okay, so this is right. Okay, so this is the sympathetic nervous system. This is part of the autonomic nervous system that kind of speeds up the heart rate, for example. So this prevents the sympathetic nervous system, so it could be used to treat tachycardia, because tachycardia is a high heart rate. Could it also be used to treat high blood pressure? Well, I'm not sure. Maybe, but I don't really know. But there's no option which is just 2. So I think 2 and 3 would be the correct answer. Muscle fatigue. You know, muscles aren't really controlled. Well, skeletal muscles to move your body aren't controlled by the sympathetic nervous system. They're controlled by the they're not controlled by the autonomic nervous system. They're controlled by the motor nervous system. Conscious, what is it called? Can't remember. Okay, moving on to the longer answer questions. Okay, so this question 16. Gestational diabetes is a medical condition that affects pregnant women. It results in high levels of glucose in the blood, even though the woman produces normal levels of insulin. So this is one of those application questions where you get given some new information about a sort of disease that is sort of related to or similar to something that you do know about, which is diabetes. So it's most similar to type 2 because insulin is produced and potentially insulin sensitivity is reduced, I expect. So there's two ways a woman with gestational diabetes can manage her condition. Okay, well, we could have diet and exercise. Well, I mean, I guess that's two ways, isn't it? Diet and exercise. But maybe if I was answering, I wouldn't just say diet. I'd say controlling diet, avoiding high sugary foods, and doing regular exercise. Let's look at the mark scheme and see what they say. So for the first question there, type 2 gets you 1 mark. Just pinch in on this, zoom in. So the first question there, yes, type 2 gets you 1 mark, this one. And then the explanation, insulin is still produced. You could have said beta cells still working. Some idea that there's less responsiveness or less sensitivity to insulin. Yeah. And the second one, diet. Now let's see here. You do need to just, have some explanation of diet, not just diet. Low carbohydrate diet, low sugar diet, regulate, control, reduce sugar or starch diet. Ignore low fat. So you do actually have to talk about sugar. So low sugar diet is the key. Exercise, manage weight gain. I guess you could have said drugs to control glucose levels. Metformin. This is a type 2 diabetes drug, this one, metformin, not injecting insulin because that wouldn't work. Okay, and we can see that the next one, is liver tissue. So B, skeletal muscle is one of the main tissues where glucose is removed. Name the other. Yeah, liver tissue, because in the liver we take glucose and convert it into glycogen. Explain why glucose is required for the contraction of skeletal muscle. So I think we start from this point. So 3 marker. So first of all, we say contraction requires ATP. And maybe we could even talk about the cross-bridge cycle for the actin and myosin. The movement of the myosin heads along the actin filament requires ATP. ATP is produced by respiration, and glucose is required to produce ATP. So those are the points that you want to make. Let's look at the mark scheme. Okay, so glucose is used for respiration. Yes, that's the obvious mark. To produce ATP, another mark. And then we needed to say this ATP needed in the cross-bridge cycle. Now, I don't even know if I would have actually got that, because you need to be, it says ignore power stroke actually. So actually, I'm not sure if I would have got that 3rd mark. I need to be more specific. You need to say that ATP is needed to break the cross bridges between myosin and actin. It's not sufficient just to say that ATP is needed for the cross bridge cycle. You actually have to say in what specific part. So ATP needed for the myosin heads to break the cross bridges with the actin filament. Note to self, be more specific. Or you could have said for the active transport of calcium ions back into the sarcoplasmic reticulum. Okay, moving back to the paper. C. During late pregnancy, women find ventilation more difficult, as the developing fetus reduces the volume of the thorax. Basically, the baby is kind of squishing your lungs at this late stage of pregnancy. This can lead to tiredness and difficulty breathing. A student uses a spirometer to measure ventilation in a woman who's 36 weeks pregnant. So this is very late stage. So 40 weeks is birth, basically. So 36 is the final 4 weeks of pregnancy. Here's the trace produced. Mean oxygen uptake at rest in women is around 0.02 decimeters cubed per second. Using these data, the student made the following conclusions. My data show that being pregnant reduces rate of oxygen uptake by 20%. Well, we need to calculate the rate of oxygen uptake here. Okay, so let's start by calculating the rate of oxygen uptake. Now, this is an evaluate question, and most of the time when it's this kind of question, the student makes a conclusion. The conclusion is wrong. So probably what they've done is they've included some sort of anomalous data. So I think that this value up here, I'll just highlight actually, is quite clearly anomalous. And if you kind of look at this, it's gonna throw you. So a much smoother sort of gradient is given down the bottom. So I think we would go with this value to this value. That final one you can't see. So let's pinch in even more and look at those values. In fact, I'm going to get rid of the... So this value here is bang on 2. Okay, so that's quite useful. 2, and it's at bang on, this is 10, this is 8, this is 6 seconds. Okay, so we can have 2 dm cubed at 6 seconds. This value here is the next one I want to go for. Now that's at 78 seconds. And the value there is, I would say, probably 1.2. I'd say that's 1.2, 1.3. Yeah, I'd say 1.2. 1.2 gm cubed at 78 seconds. So now I'll zoom out a little, and we can use that to calculate the rate. So, rate. So 2 dm cubed to 1.2 dm cubed equals a change of 0.8 dm cubed. And the time has gone from 6 to 78 seconds. 6 to 78 seconds. Which is going to be, what is that? That's 72 seconds. Okay, so 0.8 dm cubed divided by 72 seconds is going to give us our rate. So 0.8 divided by 72 gives me 0.011 dm cubed per second. Now, is that a 20% reduction versus that? The answer is no, it's more, it's almost half. So if we actually wanted to work out the percentage reduction, going from 0.02 to 0.0011 percentage, then this is how we do it. So the percentage reduction equals the change divided by the start value times 100 to make it percent. So the change is the difference between 0.02, 0.02 minus 0.011 or 0.009 divided by the start value, which is 0.020 times 100 to make a percent. It's going to be roughly 50%, so divided by 0.020 equals times 100. Yeah, it's 45% I've got. So I would imagine that that will give us 2 marks, potentially even 3 marks, but you need to write a sentence saying the conclusion is incorrect. In actual fact, the reduction is greater, around 45%. Perhaps the student looked at the anomalous first few breaths, or looked at the maximum I'd say something like that. Or look to the maximum exhale point, which is here, as opposed to the maximum inhale point, which is here. Actually, these points down here represent when the air is being inhaled out of the spirometer, and these points up here represent when the air is being blown into the spirometer and exhaled into it. So I think that would give you the marks. Let's look at the mark scheme. Okay, calculated the rate of oxygen uptake. Yes, I got that. It was about 0.11, so that's there. Calculated reduction in rate of oxygen uptake between 10 and 50, so I got 45%. So yes, that's there. And then sort of explaining it. So the claim is incorrect. A comparison of calculated rate with the statement. So they said 20%, I said 45%, so I think I would have got that mark. And validity statements. I don't know, I think, yeah, I didn't get these ones, but I would have got 3, I think. Validity statements, 1 woman, reading is not enough. 36 weeks pregnant, late pregnancy is not representative whole pregnancy. Okay, so other stuff I could have said. Yeah, okay. These are all good points here, these validity statements that I kind of missed out. Also worth extra marks. Okay, let's go back. Yeah, I should have seen that. It says, my data shows that being pregnant reduces rate of oxygen uptake by 20%. Yeah, clearly, you know, we're only looking at 1 woman who's 36 weeks pregnant, which is right at the end. So that's not enough data. Always remember those points. Let's keep going. All right, Chromista are photosynthetic protists that live in water. Chromista are different from other photosynthetic organisms because they contain a pigment chlorophyll C. Now remember that we just always be thinking, what do I know about stuff related to this? Remember, in normal plants, we learn about chlorophyll A and B. We know that chlorophyll A is the reaction center pigment and chlorophyll B is a accessory pigment. So this is what we need to talk about here. Photosynthetic pigments in photosynthesis. We need to talk about you know, harvesting photons. We need to talk about reaction center pigments, chlorophyll A. We need to talk about accessory pigments, catching you know, photons of different range of wavelengths, allowing more energy to be harvested. And we also want to talk about ultimately what happens is that we use the energy to excite electrons and then reduce coenzymes like NAD. Okay, so all that stuff is going to be worth marks. Let's have a look at the mark scheme and see how you could have got 4 out of 4. Okay, so pigments absorb light or photons. Electrons excited. Yeah, talking about electrons. And then we're talking about the different types of pigment. Accessory pigments, passing to reaction centers. So this is kind of 1 mark for all this stuff. Primary pigments lose electrons, pass electrons to ETC. I think this would have been all right. Reduced NADP. It says allow over here. For the light-dependent reaction or photophosphorylation. Okay, so pause that mark scheme there if you need to have another look. But I think that plenty of opportunity to get 4 marks there. Right, now I think it's going to get more application-y. The wavelengths of light absorbed by chlorophyll C are different from those wavelengths absorbed by chlorophyll A and B. Suggest why Chromista need pigments that are different from those of other photosynthetic pigments. Well, this is where you have to go back, because you're not supposed to know about Chromista. You've only just been told about it. So if it asks you, suggest why Chromista, you're not trying to remember something about Chromista. You've never learnt about it before. Go back to the question. What does it tell you about Chromista? The only thing that it tells you is this. Chromista are photosynthetic protists that live in water. Well, is it something about the fact that it's a protist? I don't know. Don't think so. How about that it lives in water? Well, that could be relevant, because as you go down in water, light is absorbed, and the quality of the light changes. So that's the thing. Okay, it lives in water. Water absorbs light photons of certain wavelengths, so it's going to need to make use of slightly other different wavelengths of light. And that's what it says in the mark scheme. Idea, they have to absorb light of shorter wavelengths. Well, this idea, I think we would have got idea that some wavelengths of light don't reach the depths. Yeah, so when you go down, if you have ever snorkeled or even scuba dived, you may have noticed that it gets quite blue when you go down in the bottom of the sea, and that's because the red light is absorbed. Right, here's a diagram of the Chromista cell. Oh, sorry, a diagram of a chloroplast found in a Chromista cell. So this is just the chloroplast, not the cell. I think that's an easy mistake to make. So this is the chloroplast. Outline the structural difference between the Chromista chloroplast and the chloroplast found in flowering plants. Okay, well, thylakoids stacked in threes, that's no good. So a granum with 3, that's quite a sort of small stack. In photosynthetic plants, the thylakoid might contain up to 100 of these in a granum, so that's one big difference. This says external membrane. It doesn't say 2. It doesn't say double membrane, although it does have 2 lines. That's a bit weird. Also, the other thing is there are no intergranal lamellae, because normally in a photosynthetic plant chloroplast, you get these intergranal lamellae. And DNA, yeah, that would be there. I don't know what else we're not seeing. I don't know where. We don't see any. Are there any ribosomes in there? I don't know. It's difficult to see here. I've only got those 2 points. Oh, it's only worth 2 points, so that's probably fine. I was looking for a third. What does the mark scheme say? Fewer thylakoids, yes. Chromista has no intergranal lamellae, yes. What was the other thing? Okay, plant chloroplasts have thylakoids in groups of more than 3. Yeah, that's kind of what we said already. Starch grains. I knew there was something else there. Starch grains. Plant chloroplasts have starch grains, whereas Chromista don't seem to have starch grains. Okay, C. 17.2 is a diagram of part of the plasma membrane of a Chromista cell. State and explain how one property of region A contributes to the stability of the plasma membrane. Okay, well, the property of region A, these are the hydrophobic tails. So that's it really, hydrophobic, fatty acid tails, and the property, the hydrophobic property means that they clump together, trying to get away from any water in an aqueous environment. So they will align next to each other in the absence of water in an aqueous environment. They'll stick together by hydrophobic interactions, and this will keep the membrane held together. Okay? So that's how the property works. Let's just look at this other question here before we go to the mark scheme. Ooh, this is a great one. Okay, this is such a good one, and often it throws people because it says there are different roles between the plasma membrane and membranes within cells, and this is asking about within cells. So what about within cells? So we've got it's only 2 marks here. You could have 6 marks on this really. We've got compartmentalization, which means membranes divide up the cell into different parts. We've also got sites for reactions. For example, we've got the rough ER, where protein synthesis can occur. We've also, we also talk about the Golgi body, kind of provides compartments for the modification of proteins. We can even talk about the nuclear envelope, which separates the nucleus from the rest of the cell. There's loads of stuff we could talk about. So let's have a look at the mark scheme for those 2 points, those 2 questions. Okay, so the first one, property, hydrophobic, that's the property, and the explanation, to form a bilayer or to separate two aqueous regions. Yeah, I think we got that. Oh, this is interesting. So in the diagram, I wouldn't have put this because the diagram didn't show any cholesterol, but you could have got a mark for saying that there is cholesterol in there, and that regulates the fluidity. That's interesting. The part about within cells, compartmentalization is a mark. Purpose of need for compartment separation. Sites of chemical reaction, okay, we said that. So for example, protein synthesis. Provide attachment sites for enzymes or pigments, that's an interesting one. Allows formation of concentration gradients. So this would have been marked point 1 and 2. Vesicles for transport, if we talked about the Golgi, you would have got 2 marks for that. Okay, so pause the video there if you need to look at the mark scheme a bit more, and let's crack on. Okay, question 18. Plants lose water by transpiration. The rate of transpiration varies between different species of plant. The rate of transpiration can be measured by using a photometer. Plan an investigation, here's the question, into the rate of transpiration in 2 species of plant that would allow valid data to be collected, and details of how to set up the photometer are not required. Okay, so there's a few important things here. So we're going to be using a photometer. We need to look at 2 species of plant, and we need to make sure that we can collect valid data. And then it also says that we don't need to talk about how to set up the photometer. However, we might need to talk about how to use the photometer or how to read it. Okay, so that's information there, which is going to help us plan. All right, well, let's kind of talk about how we go about planning this. You could plan this on a piece of blank paper if you had this in an exam. Or in the margin. I'm just going to plan it here. So the independent variable, the IV, is equal to the 2 species of plant, or the species of plant. So it's important that we say that. The dependent variable is going to be the rate of transpiration. And we also want to talk about how we're going to measure this dependent variable. So how do we measure? Well, we have the bubble. And it's going to sort of move along the capillary tube. We're also going to want to say that we can use that to calculate the volume. So we want to do a volume calculation. So that's going to be the L, which is the distance moved along the tube, times pi r squared, where r is the radius of the internal diameter of the capillary tube, which will be quite small, something like 0.5 millimeter possibly. So that's going to give us the volume, and then in terms of a rate, we have to divide it by a time. We have to use a stopwatch to measure a certain time, let's say 5 minutes. Look at the bubble, start the stopwatch, 5 minutes later, where did the bubble get to? So that's the dependent variable. There's also going to be a lot of marks here for talking about control variables, aren't there? If we're just trying to look at the 2 species, the control variables are very important. So what things might affect the rate of transpiration? Well, we've got humidity in the room. We've got light intensity. We've got temperature. And we've got wind speed. Okay, so if we can get an answer that kind of covers all those different things, then we're on the right track. Okay. So 2 things for you. I've got, first of all, the mark scheme, which let's go through, and then I've got a model answer, which you could even mark as well or compare to yours to see how you think you did. So first of all, let's look at the mark scheme. Here we are. Okay, so this mark scheme actually doesn't give us a huge amount of points. It's more the level descriptors that we should look at. So let's start actually with a sort of 1 to 2 mark answer. Response is aware of how to plan a valid investigation. Aware of how to plan. That's pretty sort of weak. For the level 3 or 4 answer, we need to have a detailed plan of how to carry out a valid investigation into the rate of transpiration. And for this detailed plan, you need a explanation and sequence of what to do. And as well as an appreciation of the need to obtain valid data, and it needs to be well explained. For the 5 to 6 mark answer, there needs to be a well-developed plan and sequence of what to do, and sort of a clear appreciation of the need to obtain valid data. In terms of the actual sort of points that it's looking for, we need to have a method, the movement of bubble moving along. We could have also done a mass pedometer, so that's another way, measuring the mass loss of water, timing the distance traveled by a bubble, repeating the investigation, repetition to gain replicates, and then calculation of the mean. We could even talk about a statistical test. That's an actually good one. In my model answer, I didn't talk about this. I just thought about that. Statistical test to test for difference. You could possibly do a t-test to see if the two averages are statistically different. Name variables, humidity, light, wind movement, surface area of leaves, and then some idea of how to control the variables, okay? So that's the mark scheme. Use that to mark your own answer, and don't forget to give yourself an EBI. Now let's have a look at my model answer, which I think would get 5 or 6, although I didn't talk about t-test. So let's go back to the paper, and my model answer, I'll just drag it into view up here, is over here. So I'll just put that there. How do you think that would do? You can pause the video and read my model answer if you'd like there. Okay, let's move on. 18b. Plant cell walls are made of cellulose. Cellular is a polymer of beta-glucose. Give 3 properties of cellulose that make it suitable as the basis of plant cell walls. Okay, so it has a high tensile strength. Oops. Don't just say strong. I think that's too vague. High tensile strength. It is, gosh, I'm struggling on this one. It's somewhat flexible, is it? And it is cellulose fibers are permeable to water. So water can pass through the cell wall. Let's check the mark scheme now. I'm not sure if I've got those 100% correct. High tensile strength, yes, I got that. Flexible, yes, got that. Ignore permeable, okay, so that's not what they were looking for. Only got 2 out of 3 for that one. Here are the things that I forgot. Insoluble, unreactive, inert, and can form hydrogen bonds with neighboring chains. So the fact that it's insoluble means that I guess that it doesn't just dissolve in water and float away. You wouldn't want to build a cell wall out of something that could just be dissolved. It's unreactive, which means it won't react with substances coming into the cell, I guess. And the hydrogen bond thing, it kind of is one of the reasons why it has a high tensile strength. But notice that you did need to have this tensile strength, not just strong. It's not good enough. Right, so I only got 2 out of 3 for that. Something I have to refresh my memory on. C. Cellulose cannot be digested by animals. Some mammals have bacteria in their stomachs that produce enzymes that can digest cellulose. Okay, well this is just one of these intracellular, extracellular enzymes questions, and the answer here is extracellular. Why is this the case? You have to explain. You don't just get the mark for saying extracellular. Well, think about it. The bacteria, if they were digesting the cellulose intracellularly, then they would eat all the cellulose, and it wouldn't it wouldn't help the mammals. So what's happening is the bacteria are living in the intestine. They are releasing enzymes into the intestine, digesting the cellulose, and that means that then those mammals can take in the beta-glucose that has been digested by the bacteria. So they are extracellular enzymes. Let's look at the mark scheme. So extracellular, it takes place outside of cells. Cellulose also cannot enter the bacteria. That's also something I could have said, because cellulose fibers are very, very long, and therefore you wouldn't expect a bacteria to be able to kind of take that in. And in fact, bacteria don't really take things in by endocytosis or phagocytosis or anything like that. They don't have that ability. So the enzymes must leave the bacteria to have an action. Okay, moving on. Ooh, this one. Okay, so this question. If you're in my class, I think we've seen this just recently. Perhaps you recognize this question. It's about honeypot ants even. This is an ant that carries around basically a food storage vessel on its abdomen. So here we have some data on these ants, Camponotus, Melliphorus, and Cataglyphis ants. And we have the data about the CO2 produced and the oxygen consumed. So the question asks us to work out, well, it asks us to sort of figure out what each ant diet is. So we can do this by calculating the RQ value, respiratory quotient value. And the RQ value is equal to the CO2 produced divided by the oxygen consumed. So therefore, let's do this. Let's do this for each one. So the top one is going to be 0.89 divided by 0.88. So that's pretty much 1. The next one, 0.59 divided by 0.66 equals 0.89. And the next one, 1.01 divided by 1.47 is going to be 0.68. So we're going to use these numbers below. So I would actually just go ahead and write in the RQs in the justification. So for Camponotus, the RQ is 1, just pretty much 1, close to 1. For Melliphorus, the RQ is close to 0.89, close enough to 0.9, and for Cataglyphis, the RQ is close to 0.7. Okay, so what do these mean? These are some values that you just need to know. So for Melliphorus, you would expect that it is respiring protein. Because the RQ is around about 0.9, and this is the typical RQ value for proteins. For Cataglyphis, you would expect mainly lipid, because the RQ value is around about 0.7. Okay, so those are the answers. Let's just double check the mark scheme. Here we go. So protein and 0.9, and lipid and 0.7. There we go. 3 marks for the correctly completed table. Okay, moving on to part B. It says chitin is a polysaccharide found in insects. It's used to form the hard outer casing of their bodies, the exoskeleton basically. So here's the chemical structure of chitin, and the question asks us to compare chitin and glycogen. Okay, so that's important. Chitin versus glycogen. At first glance, I thought it said chitin and cellulose because this structure looks quite similar to cellulose. But no, chitin versus glycogen. Okay, so what are the similarities? Well, there's actually a few similarities. First of all, similarity 1, we could say that they're both polymers. That would probably get us a mark. Or even better, both polymers of hexose sugars. Okay, this is a hexose. 1, 2, 3, 4, 5, 6. Hexose sugars. Similarity 2, they both contain glycosidic bonds, or they're both linked with glycosidic bonds. And in fact, it is a 1,4 glycosidic bond. So differences between this and glycogen. There are difference 1. This is beta glucose in chitin versus alpha glucose in glycogen. And also, there are no branches in chitin. And you could also say there's no 1,6 bonds that are responsible for those branches. Let's look at the mark scheme just to double check. So here we have it, pretty much exactly what I said. So they're both polymers or polysaccharides. They have 6 carbon or C6 sugars, 1 4 glycosidic bonds, didn't say this one. Differences, chitin has beta glycosidic bonds or beta glucoses. Chitin contains nitrogen. That's the obvious one, actually. I didn't even say that. It's very, very obvious. It's right there, the NH group. Chitin has nitrogen. Glycogen does not. There's no 1,6 glycosidic bonds in chitin and therefore no branching. Okay. Moving on. Okay. Here we have a 6-mark question about ATP. It says, insects use glucose to generate ATP. Outline the process involved in the generation of ATP through chemiosmosis. Now I'm throwing in a GIF here of ATP synthase and chemiosmosis. Here it is. So we can refer to it as I'm explaining the question. So I think when we're planning this question, we should think about a couple of things. We should sort of think about the location of the process. We should think about the actual process. That's probably going to be the bulk of the marks. And I think we should, I guess, also think about the kind of, maybe the sort of the raw materials, the reactants and products. Okay, 6 marks. I want to have a couple of points under each thing. Okay, so first of all, the location. Where is this taking place? Well, this is taking place in the mitochondria. And it's taking place on the cristae, which is the folded inner mitochondrial membrane. And we're also talking about protons flowing from the intermembrane space into the matrix. Okay, what about the process? We've already talked about protons flowing. So first of all, I think it might be worth just mentioning the electron transport chain. I don't know if it would get a mark, but just mentioning the electron transport chain, oxidation of NAD, plus FAD, and using that to pump protons into the, I'm just going to write the IMS for intermembrane space. So then we have a gradient called a chemiosmotic gradient. We can also call that the proton motive force. So you can see in the GIF here, these little yellow dots that are whizzing about are the protons. And there's way, way, way more protons in the intermembrane space than there are in the matrix. And in fact, I believe that pH difference is about a half pH difference. So I think that's about, or is it 2.5? I think it's about 500 times, 500 times more protons in the intermembrane space than the matrix. So that's quite a lot. So I need to put more about this process. So they have protons flow down this channel. You can see that happening sort of here. And then I'm going to kind of extend this a little bit because protons flow down the channel. It's a hydrophilic channel. And as they do so, they cause the ATP synthase molecule. That's the first time I've said that. ATP synthase, very important. Turns or twists. And this turn actually releases ATP from the molecule. and this is getting, I think maybe this teeny bit beyond A level, but basically the turn actually releases the ATP. The ADP and PI spontaneously come in and join and get stuck to the head of the ATP synthase, and it's the turn that flings it off. So we could say ADP plus PI join to make ATP. Raw materials, I think I've kind of already covered it, but I just wanted to briefly talk about NAD and FAD, and also ADP plus PI. Okay, that is definitely going to get us 6 marks. So let's have a look at the mark scheme, just to absolutely make sure, and for you to be able to mark your answer. Okay, so again, let's briefly look at the level descriptors. Let's start with the 3 and 4, because hopefully most of you are hitting this. 3 and 4 marks is a detailed description of the processes involved in chemiosmosis. Accurate descriptions of the processes. Okay, now for the 5 and 6, full and detailed description of the processes. Okay, so here, the stages, reactants, and products describing the processes. So I guess the stages involves first the generation of the gradient, the reactants involved, for example, ADP and PI and reduced NAD, and the products ATP. Okay, so let's look at all the marks that we can get. So first of all, it occurs in the mitochondria, on the inner membrane and matrix, movement of hydrogen across the membrane, use of enzyme channel protein ATP synthase, we said that, hydrogen ions are pumped out of the matrix. Did I say that? I probably would have elaborated on that a little bit more. Proton gradient created, which can be called a proton motive force. The H+ ions pass through the transmembrane protein down a channel. I talked about cristae as well. Folding of the membrane. ATP synthase produces ATP from ADP and PI. Didn't really talk about this. This is something I could have also said. The H+ ions move down their concentration gradient. And this, again, I didn't say here. Some H+ ions do leak back, so the process is not completely efficient. Okay? All right, let's go back. I'm going to pop up a model answer now. Here it is for you to have a look at, just to see how you would kind of cram all that information into a bit of text. Okay, so pause the video there if you want, and then we'll move on. Okay, let's keep going. All right, 20. A student carried out an investigation into the effect of ethanol on the permeability of cell membranes in a beetroot. The student's method comprised the following steps. Okay, this type of question immediately, I'm thinking some of these steps are going to be incorrect or they're going to miss some steps. So I want to kind of read these steps with a critical eye. Cut equal size of beetroot using a cork borer. Okay. Wash the pieces in running water. Yes, that's okay, but how many times? If you've done this practical, you should know that you need to do this, repeatedly until the water runs clear, or at least 3 times. Okay, place the pieces in 100 cm³ of different concentrations of ethanol. What concentrations? After 5 minutes, remove samples from each of the ethanol solutions. How much? How much sample? 1 ml, 2 ml? Place each of the samples into colorimeter to collect quantitative data. How? What are we measuring? Each step in the student's method relies on certain assumptions. For each of the assumptions listed below, select the numbered step from the student method that relies upon the assumption. Now, this is a weird question. Okay. Assumption A, pigment will only leak into the solution if membranes are disrupted. This relates to step 3, I believe. Assumption A relates to step 3. This is saying that the ethanol is going to disrupt the membranes and then the pigment's going to leak out. So I think this is step 3. Absorption is proportional to concentration of pigment. This is to do the absorbance of light. So this is to do with the colorimeter step. So this is step 5. Pigment will be released when the beetroot is sliced. This is to do with step 2. So when you slice the beetroot, you then have to wash to get rid of the pigment that you've released. Okay, so the student kept the ethanol solutions at constant temperature. State two other variables which need to be controlled in this investigation to ensure the data collected are valid. Well, certainly the surface area of the beetroot disc. Is going to be 1. It didn't really say that in the question because it said, sorry, in the description because it didn't say how thick the slices of beetroot were. And the other thing would be the beetroot itself, the type of beetroot. I guess there are different types of beetroot. Whether it's cooked or not, for example. That has a massive impact. I've accidentally done it with cooked beetroot once and the experiment does not work. Let's have a look at the mark scheme now and just check that we got those right and that second part. Okay, so here's the mark scheme. Okay, so 352, is that what I said? 352, 352, yeah, that's right. And then here are the variables. They've probably got a lot of different variables. So type of beetroot, yeah, that's 1. The surface area of the beetroot pieces, that's the second one. What else could we have had? Same part of beetroot, skin removed, time taken to wash the slices or number of times you wash the slices, volume of samples. These are things that we actually did label on that mark scheme. pH, that's an interesting one, hadn't thought of that. Same colorimeter filter. So we would probably test this using a blue filter to look at the red absorbance. We'd use a blue filter, I think. Okay, going back to the paper. All right, here's the graph plotted by the students showing absorbance against concentration of ethanol. 3 criticisms of the way the student has displayed the results. Okay, number 1, shouldn't be a bar chart. That is silly. We should definitely have a line graph. And second of all, I guess this would also be in the thing. 0.0 shouldn't be a category. Shouldn't be a bar / category. The scale is not the same. So here goes 1, 2, 3, 4, 5, 6 is missing. 8, 9, 1. So 0.6 is missing. Again, concentration of ethanol down here. What unit? Is it grams per dm cubed or is it percent? What is it? No unit there. And 0, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. That's okay. Arbitrary absorbance, arbitrary units, I think is okay. Okay, let's check the mark scheme, see if we missed any, but I think that was most of them. Yeah, x-axis has no units. Should be a line graph. X-axis concentration has 0.6 not included. Also no title is another one. No title. Oops. Explain how carrying out replicates would improve this investigation. So anytime we introduce replicates, we can get repeated values, identify anomalies, and calculate a mean. Okay, identify anomalies and calculate a mean being the 2 main things I imagine on the mark scheme. Here we are, yeah. So you can calculate a mean and anomalies can be identified. Also, we could have said improves repeatability and allows a statistical test to be completed. Okay, let's keep going with question 21. So this question is about the blue-ringed octopus, and I've included a photo of that here. This is one of the most venomous of all animals, and apparently one of these has enough venom to kill about 26 adults if it bites you. So you don't want to be bitten by this guy. So it bites. Its bite contains tetrodotoxin, TTX, which is a neurotoxin that can cause paralysis and death within minutes. So here's how it works. It binds to the external surface. Let's highlight a little bit here. bind to the external surface of voltage-gated sodium-ion channels. Changes the tertiary structure, the channel cannot open. Okay, so how will this affect the activity of neurons? So, well, let's think about it. First of all, the voltage-gated sodium channel would not open. So therefore, the membrane would be able to get to kind of threshold potential, but it would not then further depolarize going into the positive territory, which means the action potential would not be allowed to, would not be able to travel along the neuron. It wouldn't propagate, so therefore this would cause the neuron not to fire, basically. Let's look at the mark scheme, because I want to start moving a little bit more quickly, because I realise the video is going on a bit too long. So, sodium ions cannot enter, no depolarization of membrane, prevents the action potential going along, remains at resting potential, so the impulse is not conducted along the axon. I think the way I just described it, I think I would have got this one, and this one. That's 3, and I think that one, so that's 4. Okay, so next one. A common cause of death from TTX poisoning is suffocation, not getting enough oxygen, as a result of the paralysis of the diaphragm. So why would paralysis of the diaphragm could lead to suffocation? So if you can't if you can't contract to the diaphragm, can't contract, D for diaphragm, therefore it can't be pulled down, doesn't pull down, doesn't flatten, I'd say, doesn't draw air into lungs, therefore no lung ventilation. And then lack of oxygen and death. Mark scheme, let's check it out. Diaphragm is paralyzed, so little air drawn into lungs. I needed to be more specific here. I missed a mark. So there is no or little change in the volume of the thorax and no little change in the pressure. So you must talk about volume and pressure. I said that the diaphragm wouldn't be pulled down, and therefore the volume of the thorax does not increase, which means that air is not drawn into the lungs. Okay, TTX is also known to reduce the speed of conduction in the Purkinje fibers of the heart. What would this do to the heart rate? Suggest and explain. So the suggest part is it would decrease the heart rate. And how do I know that? Okay, well, remember, this is a very crude diagram of the heart here. We've got the sort of 2 chambers like that. We've got the septum and so on. So we've got the AVN here. Oops, I'm going to do that in blue. The AVN's here. And then we've got the action potential propagates from the AVN, which is kind of actually right in the middle there, down the septum and then up and around like this. And it's the bundle of His that carry the wave of depolarization. So therefore, this slows the heart rate because the wave of depolarization, I'll just say wave of dipole, spreads more slowly. I'm just kind of writing this down quite quick because we're going to look in the mark scheme in a second. Wave of depolarization spreads more slowly. Contraction takes longer. And therefore slower heart rate, basically. We'll quickly just look at this one here before we go to the mark scheme B. Why is transmission of action potentials along the axon slower in the absence of saltatory conduction? So this means the action potential doesn't jump from node to node, it travels smoothly along the axon, which takes more time, basically. Let's look at the mark scheme. Okay, so first of all, we get a mark for saying it slows the heart rate. So explanation 2 of any of 2 of these slows the contraction, I said, slows transmission of the impulse from the AVN to the ventricles, yes. Stops the, I think I said stops the spread, it makes the spread of depolarization slower, which I think would get that mark. Okay, and then The next bit was no nodes of Ranvier, so the whole axon needs to be depolarized. I said this. Oh no, it says ignore reference to jumping between nodes, so I wouldn't have got that mark. I need to actually say there are no nodes of Ranvier in the neurons, and therefore, wow, this is high level stuff actually. Let me explain this a little bit more, because I think very rarely in class have we talked about local currents. Okay, so very quickly, just a little detour on this. Okay, so first of all, non-myelinated, and then myelinated. Okay, so here's the membrane. We've got out, we've got in the neuron, and then in this case, we've got these nodes. So we've got this myelin sheath, and we've got the membrane sort of here. We've got out, we've got inside. So what happens during an action potential is that, let's say at this point, this is the action potential happening right here. We've got sodium flooding in. This causes a depolarization of the inside, so this briefly becomes quite positive, but then these ions move along like this. So this is called the local current. Of basically moving ions that flow along the myelin axon. This causes a depolarization over here, just enough to trigger the opening of more voltage gated channels, and then we have more sodium flowing in, more local current, more sodium flowing in, more local current, more sodium flowing in, and so on. Okay. With a saltatory conduction, we have, if we have an action potential right here at this node, we got lots of sodium flooding in. And because this is myelin here, this actually insulates the charge inside the axon, so that this flood of sodium inside here is sufficient to basically set up a current that will depolarize over here. So this is a longer local current which travels between nodes of Ranvier enough that then this one will depolarize. So this, we often say in class that it jumps from node to node, but it's a longer local current is the full explanation of how it occurs. Okay, back to the paper. Okay, we're on to question 22, just 2 more questions to go. Okay, so this question is about respiration and photosynthesis. A scientist used a respirometer to investigate the rate of respiration and photosynthesis of maize in different light intensities. So already I'm sort of thinking, well, maybe this is to do with the trade-off between those two, and maybe it's to do with this light compensation point where they kind of exactly almost cancel each other out. So here's the method. 10 maize seedlings in a respirometer in the dark for 3 hours prior to the investigation. The respirometer contains soda lime to remove any CO2. The scientists placed the seedlings in a separate respirometer without soda lime and placed it in different light intensities for 3 hours at a time. So without soda lime, place it in different light intensities for 3 hours at a time. Okay. Got you. Okay, so in this one here, this top one, this is in the dark with soda lime. Okay, so dark soda lime. So this is just the uptake of O2. Calculate the rate of oxygen uptake in the dark. Okay, so we can use pi r squared L. So pi times the diameter, look at this, diameter of the tubing was 0.1. So R is 0.05, because it's half the diameter, squared times 3.7 millimeters. And if you plug that in your calculator, we're going to get the volume taken in. So let me just do that. We've got a vigor of 0.029, so I'm going to do that, 0.5. This is millimeters cubed. But remember, this was for 3 hours. Okay, so this was 3 hours, so we want this per hour, so we have to divide this whole thing by 3, which is really not a lot. 0.0096. Which I'm going to do, sorry, 97 is 968. 0.0097 millimeters cubed per hour. That's quite a horrendous question. Let's just double check that I got that right. Yeah, 9.7 times 10 to the minus 3, or 0.0097. 3 marks for the correct answer, and if you didn't quite get there, have a look over here and see if you got 1 or 2 marks. On to the next one. Okay, 1,700 lux is a typical light intensity on a cloudy day in the UK. Calculate the percentage increase in gas production between 1,700 and 2,000 lux. Okay, so the increase in gas production. So I think we can just do 1.2 to 2.9. We're just looking at this two data here from 1.2 to 2.9. What is the percentage increase? Well, the change, we can just look at this raw number here. This is an extra 1.7. So the percentage change is going to be the 1.7 difference divided by the starting value, which is 1.2, times 100 to make it percent. So this is going to be a bigger number over 100%. 1.7 divided by 1.2, I'm sorry, 1.7 divided by 1.2 equals times 100 equals 142%. Check the mark scheme. Yep. Did it say to 2SF? No, 142 is what I got. I think that's all right. Yeah, 142 or 141.7. Yeah. Oh, sorry. Okay, back to the paper. Suggest why soda lime was not placed in the respirometer with the seedlings grown in the light. Well, soda lime absorbs CO2, and if there was no CO2, there would be no photosynthesis occurring. So this would be damaging to the plant because the Calvin cycle could not continue. Keep doing that, sorry. Okay. Removing CO2 would prevent photosynthesis. Yeah, fine. That's the mark. CO2 would be a limiting factor for photosynthesis, or Calvin cycle. Yeah, fine. Okay, the scientists made the following claim. These results suggest that in maize seedlings, the rate of photosynthesis only exceeds the rate of respiration when the light intensity is above 1,510 lux. Let's double check. Let's go back to this. Okay, so this is the value that they're talking about, this value here. So at this point, there is no gas being produced. So let's just, I think this actually is one of the rare occasions where this is a good claim. So at 1510 lux, and you do have to say lux, no movement of the bubble, or no movement of bubble, so therefore no net production of gas. So therefore, the rate of respiration equals photosynthesis, or equals the rate of photosynthesis. Now, I had to pause this video and think quite hard about this, and I actually think that the exam board have done a bit of a weird question. I'm not sure if this is actually correct because, and you know, feel free to get back at me, OCR, if you think I'm wrong. But I think, and this is kind of a side, so you know, feel free to skip past this bit if you want. But I think if you had a seedling in a sealed jar like this that was photosynthesizing, I think, remember there's no soda lime in here, so I think the seedling, if it is photosynthesizing, it'd be taking in CO2 and putting out O2 just from photosynthesis at the same rate. So I don't think photosynthesis would actually cause the volume of gas in there to change at all, and we'd just be looking at the consumption of oxygen. Well, I don't think the volume I don't think it would just change at all because there's no soda lime in there, basically. Right, moving on. All right, question 23. This is where you need the insert. I've just put that over here on the right so we can have a look at it. The student looked at slides at different tissues under a light microscope. Basically, what are they? So let's have a look at them all. Okay, well, X is an obvious one. I think it stands out for me. X is the islets. This is pancreas. And here we can see an islet of Langerhans. These are the acini around here. So this is the endocrine tissue in here. And this is the exocrine tissue out here. Okay, remember all that. Right, what are the rest? Well, here we've got two types of muscle. Y, to me, looks to be skeletal muscle. We can see these parallel muscle fibers, the nuclei kind of pushed to the outside, and we can see the stripiness of it, very striated. And the other one asks us for W. Let's just pinch in a little bit so I can see that more clearly. Okay, well, this is definitely a blood vessel. And I think, based around what tissues you need to be able to identify, I'd have to say this is probably the liver. I think this is the central vein. And this is like a sinusoid here, I think. And this is the hepatocytes around. That's the only thing I can think of. Z looks to be a type of muscle, but I think this is cardiac muscle. We can just about see the intercalated discs there. So I think that's cardiac muscle. Okay, so let's check that. I'm saying Y liver, X pancreas. Sorry, Y skeletal muscle. Why am I saying it in that order? W liver, X pancreas, Y skeletal muscle. Let's check the mark scheme. W liver, X pancreas, Y skeletal muscle. Great, yeah, good. Okay, moving on to the next bit. The student wrote about control of heart rate. We've got all that text there. Identify and correct any biological errors in the student's summary. This is a nice question. So let's first of all highlight the errors. I'm going to highlight them in pink. When the heart rate is too low, the level of carbolic acid is not in the blood. Carboxylic acid is not in the blood. Becomes higher than normal. This would be, I think this is talking about the hydrogen carbonate. So we'll come on to that in a minute. The vagus nerve sends axon potentials to the AVN. That's wrong. The vagus nerve is part of the parasympathetic nervous system, so that would slow down. To increase the contraction rate of the heart muscle, the baroreceptors in the walls of the blood vessels then detect that the pH of the blood is normal. No, okay, so that's the baroreceptors. There's not baroreceptors, so heart rate can return to resting. The endocrine system can also change the heart rate, release the hormone adrenaline from the adrenal medulla. That is correct. Causes the smooth muscle, that's wrong, of the heart to contract more frequently. Okay, so there's loads of things you could have said here, loads of things you could have corrected. Okay, so first of all, carboxylic acid. Let's just label these 1, 2, 3, and 4. So for point 1, we want to correct that to HCO3 minus, the hydrogen carbonate. For point 2, we'd want to correct that and say it's actually the accelerans nerve that is going to increase the contraction rate of the heart muscle. And in fact, it's not even, yeah, I suppose it increased the rate of contractions of the heart muscle. Yeah, I suppose that's all right. 3, it's not the baroreceptors that detect the pH, it's the chemoreceptors. And 4, heart is not, it's not smooth muscle in the heart, it's cardiac muscle. Okay, now let's look at the mark scheme to see if that is correct. Zoom out there a second. Okay, so yeah, carboxylic acid should be, they've said H2CO3. That's true. Carbonic acid, yeah, which then dissociates into HCO3-. I'm not sure if you would have got what I put, which was that. Accelerans nerve is correct. I missed this one. I'll go back and check that. Chemoreceptors, yes. Or you could change baroreceptors to pressure. Smooth muscles should be cardiac muscle, yes. Where was that AVN? Let's go have a look at that because I missed that one. Oh yes, okay, send action potentials to this should be SAN, that is the place where the nerves join to modulate the heart rate. Remember, the AVN comes later, so it goes SAN, across the atria to the AVN, down the bundle of hairs, up the Purkinje fibers. Okay, moving on to part C. Reflex actions are rapid responses that protect the body from harm. The Morrow reflex is found in babies up to 5 months of age. I've got one of those. And occurs when the baby feels its head is suddenly no longer supported. The Morrow reflex is made up of the following responses. The baby spreads out its arms and then brings them together rapidly, and the baby cries. I don't try this with my baby, but occasionally if the baby feels a bit startled, she goes like this. And that is the Morrow reflex, okay? It's kind of funny looking. So the Morrow reflex helps kind of brace babies for impact, I suppose. It protects the head. Arms out to kind of protect from falling, I guess. And crying alerts adults or parents or whatever, for help, basically. The Morrow reflex gradually disappears and usually stops completely after babies reach 9 months, and other reflexes develop as children grow older. Describe a reflex response a 3-year-old child would make to an object moving towards their eyes and explain the advantage of this response. So they would, well, I guess it depends upon the size of the response, but they would blink, they'd close eyelids. They might even duck their head, and this would prevent damage to the sensitive eye/cornea, and may even avoid the object entirely if we're talking about ducking. So let's just check the mark scheme for that final little bit. So, okay, so it allows babies to try to hold on or grasp. I don't know about that one, but okay, that's what the mark scheme says, so we'll have to go with that. But crying, I got that one. Crying draws attention to the baby, so it alerts parents. Someone encourages someone to pick the baby up. Okay, the description here was looking for blinking or shutting or closing of the eyes. Prevents damage to objects entering the eyes, or protects the eyes or the tissue around the eyes. And the explanation is that it's involuntary. I didn't say that. I need to say that it's an involuntary action. So I think they only would have got 2 out of 3 for that one. Yeah. Okay, so that is the full paper. I hope you found that useful. Have a little look, pick out the top 3 or 4 questions where you really didn't do so well, and use those questions to focus your revision on what you need to do. Okay, hope you enjoyed it, and I'll see you next time.